Doctors Longo and Roberts discussed the current status of gene therapies in rare neuromuscular disorders in this 8-part podcast series. This is derived from the symposium that was presented at World Symposium 2025 in San Diego, California on February 4 through 7, 2025, and is intended for healthcare professionals only. This podcast includes information about investigational compounds that do not yet have a regulatory approval or authorization for a specific indication. The safety and efficacy of the agents under investigation have not been established, and contents of this podcast shall not be used in any manner to directly or indirectly promote or sell the product for unapproved uses. The views, thoughts, and opinions expressed in this presentation belong solely to the author and are subject to change without notice. The contents of this presentation do not constitute an endorsement of any product or indication by Astellas. In this part, Dr. Longo will discuss ongoing gene therapies in lysosomal disorders.
I'm going to discuss some example of ongoing gene therapy for lysosomal disorder. So there are gene therapy in development for both Fabry disease, and some of these involve ex vivo gene therapy. Many others involve systemic administration with an AAV, Gaucher disease type 1 that affect the periphery, and Gaucher disease type 2 where the replacement should occur within the central nervous system because this condition affects the brain. And then there is already one approved gene therapy for lysosomal disorder, which is for the early onset metachromatic leukodystrophy, and this has been approved both in Europe and now even in the United States, which consists of ex vivo gene therapy with the administration of an extra gene that restores the function of the defective enzyme. And now there are many others that are ongoing for the same indication. There are gene therapy programs for GM1 and GM2 gangliosidosis, and at least one for Krabbe disease. And it is important to know that some of these conditions are actually included in the recommended uniform newborn screening panel. So basically, we would have access to patients in a timely manner for some of these conditions. Then there are several gene therapy under development for the mucopolysaccharidosis, including MPS 1, MPS 2, MPS 3A, MPS 4. And then there are different types of lysosomal disorder. The one caused by mutation, integral membrane protein, not enzyme within the lysosome, but protein that are present on the membrane of the lysosome. And this gene therapy that are being tested, it is for cystinosis, that it is caused by a defective lysosomal transporter, and for Danon disease, which is caused by a deficiency of an integral membrane protein. Finally, one lysosomal disorder, which obviously seems a metabolic condition, but it is really not, is glycogen storage disease type 2, or Pompe disease, in which there is the intralysosomal accumulation of glycogen. And there are several ongoing clinical trials to try to correct the problem in this condition. Now, I'm going to discuss some of the most advanced programs in lysosomal storage disorder. This includes one for Fabry, which is on an accelerated approval pathway with phase 1 and 2 data, one for Gaucher disease type 1, and obviously I'm going to discuss the one that has been already approved for metachromatic leukodystrophy. There is one for Hunter syndrome, and the difference of the one for Hunter syndrome, it is an example of the direct administration of gene therapy within the central nervous system. And finally, there is one ongoing for glycogen storage disease type 2, or Pompe disease in adult patients. Now, in gene therapy for metachromatic leukodystrophy was the first gene therapy approved for a lysosomal disorder in humans. And this requires harvesting the CD34 cell from affected patient, and then introducing the aryl sulfatase A gene back in this cell, and then placing them back inside the patient again. This has been very effective in patients who were treated early, and obviously the treatment needs to occur before there is irreversible brain damage in this patient. And one of the things to keep in mind that all of the gene therapy that we are being tested will require prolonged surveillance of patients receiving it. And all of the program usually include about 15 years of follow-up of these patients to make sure that there are no unwanted side effects that are not evident immediately. Now there are currently a phase 3 clinical trial for the late juvenile form of metachromatic leukodystrophy that it is enrolling, try to expand the indication of gene therapy to people who have a milder form of the disease. There are gene therapy for Fabry and Gaucher. So the previous one was ex vivo lentiviral gene therapy. The one for Fabry and Gaucher, they are systemically administered, the one that I'm going to discuss, and they involve adeno-associated viruses. Usually these therapies are given to stable patients that don't have a risk of deterioration, and basically the one in Fabry disease has caused a significant improvement actually in the glomerular filtration in a relatively short time. And it is obviously one needs to make sure that that continues to be the case with longer clinical trial and expanding that to a larger number of patients. The one for Gaucher disease, there have already been 2 different trials. One to identify the effective dose, the second one to look for efficacy. Many of these patients have been able to discontinue the concurrent either enzyme replacement therapy or substrate reduction therapy. And obviously they are expected to progress into the trial and possibly to provide positive results. In the case of Hunter syndrome, the difference is the adeno-associated viral vector is delivered by direct injection into the central nervous system. And what we know so far is that this therapy is effective in reducing some of the biomarkers for Hunter syndrome. In this specific case, the non-reducing end of the glycosaminoglycan that accumulate in this condition, indicating that there is at least biological activity with actual biological action in the central nervous system. And this is obviously bodes well for thinking that there might be an effect on cognition and development. And so far, the patients are relatively stable. And more importantly, many of these patients remain very stable and they seem to be doing very well. Obviously, there is not much of an alternative in this disease because the current therapy does not cross the blood-brain barrier, and this offers the opportunity to this patient for normal development. Finally, there is an ongoing clinical trial for late-onset Pompe disease. In this condition, the gene therapy is given to adults on a stable dose of enzyme replacement therapy, and many of the patients who have received this gene therapy have been able to discontinue the enzyme replacement therapy and remain stable in terms of pulmonary function and motor activity. And obviously, the fact that they've discontinued enzyme replacement therapy has impacted in a positive way their quality of life and their overall outlook.
In the next part, Doctors Roberts and Longo will discuss treatment with gene therapies.